Hi, welcome back to Physionic or a Physionic long form video. Today's video is going to be a part one of a two-part series discussing the role that fat has, nutritional fat, molecular fat that is found within our body. Those are the same thing. How fat affects cancer or cancer progression. So throughout this discussion, and I'll have a lot of different visuals for you so that you can actually see what I'm talking about as we're going through it. I'm going to give you a background on the utilization of fat or different reasons why cells in general, as well as cancer cells, especially cancer cells, use fat. Then I'm going to go into some examples of cancer's relationship with fat, and then some mechanisms of fat reliance by cancer cells. Now, in part two, then, we're going to go into a few different therapies, including nutritional therapies that can attack or can affect how cancer is able to use the mechanisms that we'll be discussing in this part of this series. So if that's interesting to you, then stick around. Now, the research that I'm going to be going over is from a particular review that comes from the British Journal of Cancer, I believe. We'll be looking at it in just a second. And this journal goes into all kinds of different research. They go into far more specifics than what I'm going to be supplying here. Of course, I'm going to have that review linked for you, as well as my notes. Now, the notes do their damnedest to distill the information that the researchers point out, because, of course, keep in mind that these are cancer researchers, so they're using all kinds of different terminology. So I've distilled some of that terminology into the margins through my notes. So if you want more details than what I provide in this video, then please hop on over there and you can see the notes for yourself. However, I do go into some pretty immense detail when it comes to this video, so hopefully you're able to learn quite a few things. We're going to really go into the weeds in terms of some of the mechanisms, the cell biology mechanisms that occur, as well as a little bit of molecular biology and maybe a little bit of physiology as well. So without further ado, let's go ahead. Well, actually, one more further ado. If you're not familiar with who I am, you may be thinking, well, what does this guy know about cancer? Granted, I am not a cancer researcher. However, I am getting a degree. Currently, I'm in my PhD, my fourth year of my PhD in molecular medicine. And we've definitely been inundated with a lot of information on cancer, because I don't know if you've heard, there's this disease out there called cancer, and people in medicine have to know about it. So take that for whatever you think it's worth. But so I do have some information that I can talk about related to cancer, and with my background in physiology and molecular and cell biology. So now, without further ado, let's go ahead and jump into this video. All right, so like I said, the initial question that we're going to be asking, or well, really, I guess the only question that we're going to be interested in, except for then in the second part, is what role does fat have in cancer? How does fat relate to cancer cells? And this information comes from this review article. Yes, I did get it right. The British Journal of Cancer. So this is a review article, meaning that they have, I don't know, something like 100 different studies that they look over, and then they put it all together into a concise format. Now, concise, of course, still being like 12, 14 pages of dense material. So it's called Reprogramming of Fatty Acids, which are fat molecules. So anytime I say fatty acids, or anytime you see this word fatty acid, it means a fat molecule, which is very similar to what you consume when you consume nutritional fat that you might find on the nutrition label on the packaging of what you eat. Fatty acid metabolism in cancer. All right, so to start things off here, let me show myself. to start things off here, I'm going to give you, like I said, a bit of background. So here we've got just a regular cell. And there are two initial ways that fats are important to cells. So cells can take up fats from the environment. So talking about what's known as the extracellular fluid. So the liquid that's outside of the cell. So like the blood system would then transfer nutrients into what's known as the interstitial fluid. And that interstitial fluid is surrounding the cell. Now, in that fluid, of course, then there's these fat molecules. These fat molecules that can then enter into the cell. Doesn't have to be a cancer cell, but as we'll be focusing in on cancer cells, that's what we'll definitely be talking about. But it enters a cell, and then it can be used for, as we'll see, a bunch of different functions. Now, the second thing that cells can do is they can convert what's known as an acetyl-CoA molecule, which comes from, and I'll show you some diagrams of this, which can come from glucose, it can come from amino acids, but ultimately they get produced or turned into an acetyl-CoA molecule. Then they get converted to a fat molecule. So fatty acid, fat molecule, both same thing. So this is de novo lipogenesis. This occurs in your liver cells as well as your fat cells. However, what's unique about cancer is that a lot of the cancer cells can do it as well, even if they come from, let's say, breast tissue or if they come from ovarian tissue. They suddenly have this ability to produce fats, unlike our regular physiologically normally functioning cells. So to get a little more granular with this, looking at the same image, again, if you've got these fat molecules that enter the cell, whatever that is, physiological or cancerous, then what can happen is one that can get stored into lipid droplets that are found within the cell itself. The second thing that can happen, however, is that they can be utilized. And this utilization is what we're really going to be focused on, because fats aren't just used for energy, which we will certainly be looking into, but they can actually also be used for signaling. They can be used for cell membranes. They can be used for a litany of different things. So once a cell takes up a fat, it will be kind of proportionally be used up depending on what the cell needs. Now, the same can be true if, let's say, a cancer cell were to produce a fat from an acetyl-CoA molecule, were to produce a fat, then it can be utilized. It can also be stored. This is common as well, but I only drew an arrow for utilization. But yes, this created fat can also be stored. Okay, so that's a bit of background on the general use of fats, like what kind of fates there are for fats. Now let's hone in on cancer specifically. All right, so to enter the cancer cell, or really to enter any cell, fats have to have these transporters. And this is a very simplistic illustration of what these transporters look like. But they have these transporters that are found on the cell membrane. So along this entire cell membrane, you have these transporters that are known as fatty acid transporters. Remember, fatty acid is the same as a fat molecule that you would find in your nutrition or that's released from your fat cells. And that second part is going to be important in just a second. So these fatty acid transporters, also known as their more numerical term, are CD36. So if you look up CD36 or fatty acid transporter, you'll learn about these in more detail. But the fats are outside of the cell in the extracellular fluid, also the interstitial fluid. However, they're different versions of almost the same thing. These fats will then enter the cell, and then we end up undergoing those fates that I just discussed: storage, utilization, etc. Now, the amount of these fatty acid transporters is much higher in certain cancers. Now, when I'm talking about these cancers, I don't want you to think that this applies to all cancer. All cancer has this. But some cancers do have this, where they have a lot more of these fatty acid transporters. Now, if you were to think about that just kind of logically, what does that then mean? It means that potentially this cancer would be more prone to using fats for a variety of different fates. It doesn't necessarily mean that it's going to use it for metabolism purposes, but it does mean that it's probably going to use fats in some way. And so they end up having more of these fatty acid transporters compared to physiological tissues. So why that's important in this scenario is, and I'm starting to get into some of these examples now, is when particular cancers, again, not all cancers, and I really want to add that stipulation because I really think that for this entire video, when I'm saying cancers, I don't want you to think that every single cancer has this hallmark, because there are going to be differences, and I'm going to point out some of those differences. But in certain cancers, like breast cancer, ovarian cancer, and there's still heterogeneity, meaning there are still differences even within those tissues. But generally, these types of cancers will be surrounded by fat depots or fat tissue found as adipocytes. So adipocytes being fat cells. So these fat cells, of course, contain the actual fat molecules that we've been talking about. So these fat molecules are found when they're not freely surrounding the cell, when they're not freely in the interstitial fluid, they're stored in these fat cells. And this is kind of basic, exchange of different fats. Now, the cancers will end up finding themselves around or growing around a lot of fat cells. And you can imagine why, if they have high levels of these fatty acid transporters, they find themselves around a lot of fat cells. Then when the fat cells release fat, those fat molecules get sucked up from the interstitial fluid that's between the fat cell and the cancer cell, gets sucked up into the cancer cell. This gives an advantage to the cancer cell in that it can constantly be fed by these fat cells. So what researchers have done is that they've co-cultured, meaning that they've taken cancer cells and adipocytes and put them on a plate together, or in some way kept them in close proximity. And what they found is that if they co-culture the cancer cells, as opposed to when the cancer cells are in isolation, still being fed with the requisite what they need to survive, that the cancer cells will migrate a lot more, meaning migration, meaning more metastasis, potential metastasis, where a cancer cell will move from the tumor itself right here to moving away from the tumor and generating another tumor somewhere else, if it can find an anchoring point to end up growing somewhere else. So this is where you get into further stages of cancer progression, stage 3, 4, etc. So it's important here in this example that this cancer cell is more potent when it is surrounded by these fat depots, by these fat tissues. So this is a great example of the uptake of fat. This is an example of this right here, the uptake of fat. Now, looking at the synthesis of fat, so de novo lipogenesis that we talked about earlier, we've got this cancer cell, and within that cancer cell, what it's doing, what any de novo lipogenesis is, or lipogenesis, is the conversion, as I briefly mentioned, of something like glucose or amino acids into a fat molecule, a saturated fat molecule. In this instance, and then something else can happen. It can be desaturated into monounsaturated fat. But for the time being, it gets converted to a saturated fat. And I'll show you the enzyme that actually does that in just a little bit. But how does it do that? What's the purpose of that? Well, one is to generate more fats, and we'll see why those fats are important a little bit later. But how it does that is through the conversion. The reason why I've got this chemical structure of a glucose molecule. So this is my animated version of a glucose molecule. And if we were to actually look at the chemical structure, these are both identical to one another, even though they look different. This is just kind of a linear representation of all this. So we've got, in this situation, a series of different carbon atoms that are stitched together. So 1, 2, well, I guess they're labeled here from right to left. 1, 2, 3, 4, 5, 6. So you've got these 6 carbon atoms that are stitched together, and what de novo lipogenesis does is it takes these carbon atoms and it converts them to a series of different other molecules to eventually convert them to what I mentioned earlier, the acetyl-CoA molecule. From there, from acetyl-CoA, then you get the conversion to fats, because fats are, and I wish that I had added another chemical structure here. If you look at the chemical structure of a fat, it's just a linear series of carbons that are just stitched together. And that's exactly what happens. So it unravels this glucose molecule and turns it into, stitches it together, a series of acetyl-CoA molecules, into a fatty acid molecule or into a fat molecule. So this is another mechanism by which cancer cells can generate the fats that they need. So now, to get a little more granular here, to look at, for example, the use of fats, again, in this situation, they're taking up fats into the cell itself. So this is the cell membrane from outside the cell into the cell. Now what I'm going to be focusing in on is this cancer cell has mitochondria, just like most of our other cells. So this cancer cell with this mitochondria, so here we've got, I'm making this as basic as possible, but here we've got our mitochondrial membrane, or one of them, the outer mitochondrial membrane, and we've got this protein that is found embedded into the mitochondrial membrane. And this protein, known as CPT1, will allow, in a really basic sense, will allow fat molecules that are inside the cell but not inside the mitochondrion, will allow these fats into the mitochondrion, and typically then they're utilized for energy, for ATP generation. Now, certain cancers have this propensity to not necessarily make more fat, but utilize more fat, so therefore focus more on the utilization of fat through an overexpression or an increase in the expression of the CPT1 gene. This is CPT1 here. This is the protein. This is the gene. So in the nucleus, where all the DNA, or most of the DNA, is kept, we've got this CPT1 gene. So what happens is that these cancers, these particular cancers that have high levels of this protein known as MYC, not Nick, not me, but MYC. I don't cause cancer unless you really, really hate these videos. Then I'm terribly sorry that I've caused cancer. But in general, I don't cause cancer. MYC, M-Y-K, M-Y-C, my apologies, MYC, MYC, will then enter the nucleus because there's high expression of this MYC protein in these particular cancer cells, will bind to the CPT1 gene and lead to its expression, meaning that it's read a lot more. As it's read, it produces a lot more of these CPT1 proteins, which are then embedded into the mitochondria, which allows more of this fat. So you're not only accepting a lot more fat into the cell, but you're also accepting a lot more fat into the mitochondria to generate energy. So this is one mechanism by which fat can be utilized by cancer cells, and that cancer cells can it's almost like they're supercharged to use these fats. If they're prone to utilizing fats, they're suddenly supercharged because they have this environment where that is constantly being propagated by this MYC protein. Now, interestingly, of course, if you were to inhibit and we'll discuss these kinds of mechanisms in the next episode, but if you were to inhibit CPT1, what's interesting is that suddenly these cancers suffer substantially. Or if you were to eliminate MYC, suddenly these cancers start to die off. So not only is this an advantage, it's also a disadvantage. Now, I can already tell the questions that I usually get around this kind of stuff if I say that it's highly prone to death if we get rid of this one protein or if we inhibit something, whatever it might be. The problem isn't doing these things. The problem is how do you get it? How do you get your drug or how do you get your intervention, nutritional intervention, whatever, to the cancer? And after you've done that, how many widespread effects are there to other tissues that also have these proteins? Because these are not unique to cancers. It's just that the amount of each is unique to this particular type of cancer. So yes, we know the mechanisms. We can do it in a cell culture plate. We can do it to animals, but actually doing it to humans and having them not suffer gruesome side effects is a whole nother story. So that's really the main issue with cancer research. Okay, so this is, again, an example of the utilization of fat. Now let me give you a more detailed explanation or understanding of how the synthesis of fats may affect cancer cell growth and division or proliferation. So now we're focusing in, so here we were looking at the mitochondrial membrane right here. Now I want to focus in on the cell membrane again, which is what we did with the CD36, the fatty acid transporters. So this cell membrane is littered with all of these types of receptors. In this example, we're talking about a HER2 receptor, which is like human epidermal receptor, I believe. So a HER2 receptor. And when these cancers have this binding of the HER2 receptor, what happens is that the HER2 receptor has two different parts and it will combine. And then you have the binding of what's known as a ligand. In this situation, you're talking about a ligand would be like a hormone or some sort of cytokine, some sort of other molecule. But you have the binding to that receptor. And then you have a series of changes that occur inside of the cell. And then you have a series of other changes. I mean, this arrow just completely summarizes a ton of different things that change. And you ultimately get then cell growth and cell division, which is, of course, then making more cancer cells. That's essentially what that means. Now, what's interesting here is that what researchers have found is that this protein that or this de novo lipogenesis that I mentioned earlier, with this acetyl-CoA being converted to a saturated fat, what's interesting is that the protein responsible for that, or one of the proteins known as fatty acid synthase, or FASN, F-A-S-N as an abbreviation, if they inhibit the function of this particular protein, they find that the cells not only no longer grow, they no longer divide, but they also end up dying. So just like if you were to inhibit MYC in these other types of cancer cells that are more focused on the utilization of fat, or if you were to inhibit CPT1, suddenly these cancers suffer significantly. The same thing is true for another grouping of cells, cancer cells, that where you inhibit the fatty acid synthase. And we're not exactly clear, it wasn't entirely clear as to how this fatty acid synthase interacts with all the different molecules, because there's a series of different molecules that get activated to end up leading to cell growth and division. But somehow this thing interacts with these molecules in such a way that that affects this overall cell growth and division, or the general health or survival of the cancer cell. So if we inhibit this, then we get a stoppage of this entire cell growth and division. Again, just examples here of how fatty acid enzymes, how fats themselves affect cancer cells. Okay, so now let me go into, I'm looking over my notes here real quick on the side to make sure I'm not missing anything. All right, so now allow me to go into how fats specifically, the actual molecule of fat. So we talked about, these different enzymes, the background on fats, if they're taken up, if fatty acid synthesis. Now let's talk about how the fat molecules themselves affect cancer progression. So in this scenario, what can happen is for cell division to occur, let me go back one slide. These membranes are largely made up of fats. So you have these proteins that are embedded into the membrane, sure, and you have other proteins as well, and you even have carbohydrates, but largely we think of them as these phospholipids, so lipids being the fat portion. So the membranes are made up of a lot of fat. So obviously these cancer cells, and really all your cells in general, need fats to then be able to proliferate, to be able to divide into two, so from one cell to division into two different cells. So it's going to suck up a lot of fats to be able to generate two separate cell membranes that are intact and separate from one another. So fats are used for cell membrane synthesis. So keep that in mind. Now, it's also advantageous for example, in cancer cells to generate their own fats because they can also generate or stop the creation of fats, specifically saturated fats. So the advantage to that is that unsaturated fats, specifically polyunsaturated fats, but even monounsaturated fats, but like I said, especially poly, meaning that you have multiple degrees of saturation. What does that mean? It means that you have a change in the chemical structure of the fat that leads to this kink right here. So the more of these kinks that you have in this fat molecule, the more prone it is to damage by reactive oxygen species, meaning these reactive oxygen species are unstable molecules. Again, if you've been following Physionic for a long time, you know all this already. But these unstable molecules will destabilize molecules around them by tearing away components of those other molecules. So this reactive oxygen species would interact with this unsaturated fat and would tear away at the kink or tear away at different components of this fat, therefore leading to what's known as peroxidation, which means that there's damage to this molecule and now it's less or non-functional, meaning the cell can no longer use this or can't use it as efficiently. Now, what's interesting is that saturated fats, although they're not completely immune to reactive oxygen species, they incur less damage from reactive oxygen species. So when cancer cells produce more of these fatty acids, they're initially producing them into saturated fat. Now, that's not unique to cancer cells. Non-cancer cells also do that. But then typically what happens is they get converted from saturated fats to a unsaturated fat. Now, cancer cells can produce large amounts of these saturated fats, which can then be used in the cell membrane to produce more of these robust cancer cells that are less prone to damaging by oxidative stress. This is oxidative stress, this ROS. So they become more protected against reactive oxygen species, specifically in the cell membrane. Okay, so another area is in cholesterol. So I wanted to throw this in here, although it's slightly different, but cholesterol is important for a number of different reasons. So it has advantages and it has disadvantages. So I don't want you to walk away from this thinking that fats are absolutely then always beneficial to cancers. They're not. And this is one example. So with cholesterol, for example, when you have cancer cells that need to move from one area of the body to another area of the body, meaning that they're trying to metastasize, the more fluid the membrane is, so think of it like your body. Think of it like if you're really stiff, it's hard for you to move. you you're aging, or you're just you wake up and you just have a stiff neck because you slept on your neck the wrong way, then it's suddenly really hard for you to move. Same thing happens to a cell membrane. I mean, it's not like the cancer cells wake up and they're like, damn, oh, I slept badly last night. They don't do that. But the analogy being that the stiffness decreases the movement ability. Now, cholesterol increases the stiffness of the membrane. This is a negative for cancer cells in that they are then less able to flexibly move through the body and move to new destinations, to metastasize, to create new tumors in other areas. So that's a negative. One of the compensatory mechanisms, and I forgot to draw an image for this, but one of the compensatory mechanisms of cancer cells is that they will then efflux cholesterol. So if they have high amounts of cholesterol, they will actually start to export cholesterol out of themselves. So they will move cholesterol from inside the cell. How we saw fats going into the cell, cancer cells will then just eject cholesterol out of themselves to reduce the cholesterol burden inside the cell, therefore reducing the effect of these cholesterols being integrated into the cell membrane itself. So this creates a more rigid cell membrane. That is a negative. However, there are positives to this as well. So if you're talking about a tumor itself that does not necessarily need to progress or need to split and create more and more cells, or for it to move to other areas of the body, if the tumor is staying more localized, cholesterol can actually act in two beneficial ways. One is because of this increased stiffness of the membrane, it will protect against anti-cancer drugs. So these anti-cancer drugs, which might normally just fluidly move across the membrane because they are membrane permeable, and that's of course probably not true for all of them, but for some of them that are membrane permeable and can just cross this cell membrane, they're suddenly not able to, because of these cholesterol groupings, that even though the membrane is more stiff, it also keeps these anti-cancer drugs at bay. So it protects the cell, it protects the tumor from being damaged by these anti-cancer drugs. Now, the second advantage is that these cholesterol groupings are also known as what are known as lipid rafts. These lipid rafts are these essentially just these groupings of cholesterols and fats and this kind of waxy substance that cholesterol is that anchors a series of different proteins in close proximity to one another. This is actually a relatively recent discovery, if I'm not mistaken. I remember learning about this back in one of my classes. And so if we take an example like the HER2 receptor that we talked about earlier, that if it's activated, it's bound by the ligand, then it combines these two components combine, and then you have the signaling that occurs for cell growth. I'll show this to you again. So right here, so this HER2, gets bound by the ligand. It can be a hormone, whatever it is, and then you have the number of different signals, and then cell growth and cell division. So with these lipid rafts, they can actually increase the density of these HER2 receptors. It doesn't have to be HER2, it can be other receptors as well. And this means that then you have more of these receptors that are closer together, and they can combine more efficiently, and therefore you have strong signaling that's occurring from these isolated sections of the cell membrane. This leads to greater signaling for cell growth, cell survival. So within a cell that it would grow. But like I said, there are still disadvantages in that the tumor, it can help the tumor itself, but it hurts the tumor in terms of spreading to another area. So hopefully I've done justice to explain that. And now the final thing that I wanted to discuss here is other ways that fats. So we talked about how fats can be used in the cell membrane, and we talked about how cholesterol can be used in the cell membrane. But now let's talk about things that are not necessarily cell membrane oriented. And fats can also be utilized for cell energy when glucose is not available, or the circumstances don't allow for glucose to be available or to be used. So there's two different signaling pathways here, and this is a drastic oversimplification of what's going to happen, of what actually happens. But in an example like omega-6 fatty acids, so this is an unsaturated fat, it can be acted on by a protein known as COX, so COX. And COX or COX will then convert an unsaturated fat like omega-6 fats into prostaglandins, which are another type of fat. They're essentially just a different version of a fat. These prostaglandins will then bind to the outside of a cancer cell. So I don't have the cancer cell shown, but here we've got our cell membrane. Again, same thing as what we've been looking at. And embedded into that membrane are, again, other receptors. So the ones that we talked about earlier, these HER receptors are known as tyrosine kinases, and these are known as GPCRs, or G-coupled protein receptors, GCPRs. It varies, but GPCRs. So these G-protein coupled receptors are another type of receptor that can be bound by a ligand, so same situation, but in this situation it's an actual fat that's binding, so a fat is binding to the exterior of the cell. This leads to a series of changes that are not depicted, and this eventually leads to a ton of cell signaling that eventually activates a protein known as ERK, or ERK, which then leads to particular genes being read, just like we did with CPT1 here. So these genes, so these proteins would enter the nucleus and read these genes and lead to the greater expression of proteins that help in cell growth, cell migration, and cell survival. Cell migration being like metastasis. Now beyond that, they can also, another example of that would be to activate things like VEGF, which is another protein that promotes the production of blood vessels. So think about that, right? If you have a tumor, let me go full screen for this real quick. If you have a tumor that's built up, let's say it's this size, and it's growing as quickly as it possibly can. It's growing faster than any other physiological system in your body. It needs nutrients. So the only way it's going to get nutrients is by building these highway systems that go into the tumor. And the highway systems are your blood system, right? Circulatory system, the cardiovascular system. So what it does is it has to release these cytokines, these molecules that attract the right cells to produce, kind of like construction workers, to produce more of these blood vessels, to create more of these highway systems into the cancer or into the tumor. Now this, as that's being produced, that's known as angiogenesis, this then allows the cancer to continue to grow because it's getting more and more highway systems. So it's taking up all of the nutrients it possibly can. Now, if it runs out, like if the highway system can't supply enough nutrients for the cancer cells, they can't grow. So it continues to throw out more of these factors like VEGF that I was talking about. So in this whole situation with prostaglandins binding the GPCR, the activation of ERK, cell growth, migration, survival, as well as angiogenesis, you can also get the expression of things like metalloproteinases, which are MMPs, which are other proteins, kind of like VEGF is a protein, CPT1 was a protein, MMPs are proteins that can be released into the surrounding environment and start to destroy the surrounding environment to create more room for these cancer cells to move. So those are metalloproteinases. So I think, to be honest, I think the very first full study that I ever covered on Physionic ever was on a paper in 2018 that covered exactly that process, how cancer cells produce these MMPs and allow for migration of the cancer cells. Really, really cool paper. So certainly check that out to look up like cancer migration Physionic. Okay, so the other mechanism, however, is when fats, saturated fats, or I think any fat, but it can also include saturated fats, can enter the cell. And of course, you'd usually have this transporter that allows for these fats to enter the cell. Remember the CD36, the fatty acid transporter. And then can be converted into what's known as DAG molecules. These DAG molecules are then another version of a fat. So it's just a conversion of just from your base fat to a different type of fat. So these DAG molecules can then be used to also stimulate a series of different intracellular communication proteins like ERK, and can also lead to cell growth, migration, and survival. So these two mechanisms that I'm describing here are two independent mechanisms by which fats directly affect cancer cell progression. Okay? So hopefully this offered some information, some use for you that now you have an understanding of how, and this is definitely a dumbed down version of what really happens. There's a lot more. I mean, I'm not saying that any of this stuff isn't true. It's true, but there's a lot more that I didn't cover or glossed over. All right, so if you're skipping to this point, I always say this, but if you're skipping to this point, I just want to say that you have missed out on so much detail that this is essentially useless. But I'm still going to do a conclusion section and a bit of a takeaway. Now, if you have ridden with me throughout this entire video and now gotten to this point, congratulations, because you actually know all the details of what this conclusion actually means. So there are many ways that fats interact with cancer. Some are advantageous, some disadvantageous, from cell signaling to changing cell membrane structure to cell survival and growth. So some examples of that would be like the reliance on fats when glucose levels are low. We talked about fat utilization for ATP generation or cell energy generation. Faster metastasis or the spreading when around fat cells. Now, again, the stipulation here is that we're not talking about all fats. We're just talking about particular fat, or not all fats. We're talking about particular cancer cells. We're not talking about all cancer cells. And another example would be that cholesterol offers protection against anti-cancer drugs and faster growth because of the localization of all those receptors we talked about. But it also, on a negative, it decreases the ability for these cancer cells to then move or spread or metastasize. All right, so that was part one. I hope that now you have a broader understanding of how fats affect these cancer cells. And I know I've said it now 2-3 times, but seriously, don't think that if you consume fat, that suddenly you will have cancer. That's not how it works. It's if you already have cancer and that cancer has a profile, an enzymatic profile, a genetic profile that lends itself to some of these advantages from fats, it's important to know that kind of stuff, because the therapies that then researchers and doctors, MDs, physicians can then use can be tailored against that particular type of cancer. But as you can see, and again, this isn't even covering everything, as you can see, there's a lot more here than what I showed. This is almost like an introduction, but certainly check out my notes where I have more information, where I tried to distill things as much as possible, but it's a complex topic. But if you're interested in the therapies, that's what I'll be covering next. So I will speak to you then. Have a great day.